This video will cover 4 different types of cell signaling within the body, starting with G-coupled protein receptor signaling, or GCPR signaling for short. A ligand binds with a receptor on the plasma membrane, causing the activation of the G-coupled protein via GTP. The protein will activate a secondary messenger, adenylyl cyclase, which will activate cAMP to carry out some cellular response. In the case of epinephrine, cAMP activates protein kinase A, which will then phosphorylate and activate phosphorylase kinase. This causes the activation of glycogen phosphorylase, the enzyme responsible for glycogen breakdown, and deactivation of glycogen synthase. The second cell signaling type covered is ion channel signaling. A ligand will bind to a chemical protein in the cell membrane, allowing for the flow of ions inside the cell. This results in the activation of a secondary messenger that will remove the phosphate groups of a transcription factor in the cytoplasm. Then, this dephosphorylated transcription factor will enter the nucleus of the cell and transcribe DNA, causing a cellular response. In the case of calcium ions, capsaicin binds to TRPV1, which will allow the facilitated diffusion of calcium ions and the activation of calcineurin. Calcineurin will dephosphorylize NFAT2, the transcription factor, that will then transcribe DNA, changing immune cell gene expression. Next, we have RTK signaling. A ligand binds to an RTK receptor on the plasma membrane, resulting in the activation of a kinase, resulting in the phosphorylation and activation of more kinases, the last of which will enter the nucleus and cause a cellular response through transcription. In the case of an epidermal growth factor, the RTK receptor will activate MAPK kinase, which will phosphorylate and activate MAPK kinase, which will phosphorylate and activate MAP kinase, which will initiate the transcription via c-MYC, expressing mitosis-inducing genes. Lastly, there's intracellular signaling. A ligand binds to an intracellular receptor. This ligand-receptor complex will then enter the nucleus and bind to genes, acting as a transcription factor. The mRNA that is then produced will create a new protein. In the case of the hormone aldosterone, the aldosterone mineralocorticoid receptor complex will diffuse into the nucleus and transcribe the mRNA that will be translated into the epithelial sodium chloride channel protein.